We're ready to go. Dr. Lewis, would you like to introduce our first speaker?
Yes, I am very excited to introduce our first speaker for today. Ms. Leah Ellman-Stortz is a master's student that's working with both Dr. Terry Gentry and I. She comes to us from Rochester, Minnesota, and she majored in natural sciences with a minor in writing during her undergraduate career at the College of St. Benedict. And interestingly enough, during this time, she worked for my office mate in graduate school, who I'm sure many of you probably remember, Dr. Joseph Storleen. He was a PhD student of Dr. Frank Hans, who also worked with Terry Gentry. She's also worked as a writing tutor and a clinical lab assistant before joining the Soil and Crop Sciences Department at Texas A&M University to take on her master's degree. So Leah, with that, I will turn it over to you.
All right, thank you very much, Dr. Lewis. Let me get this started. All right. Can everyone see this?
Well, I can't speak for everyone, but I can see it just fine.
Fantastic. All right, thank you everyone for coming. And I will begin my presentation on the influence of cover crop selection on soil microbial activity and greenhouse gas fluxes during a transitional period to organic agriculture. As the title suggests, the purpose of this study was to investigate how cover crop selection and a recent transition to organic management impacts soil health and greenhouse gas fluxes of agricultural production in West Texas. Specifically, we are focusing on soil health, which is more of a biological measure of nutrient cycling, say, according to microbial activity. So why is it important to study organic agriculture? One big reason is a massively growing demand. For example, as of 2016 alone, sales for organic products was worth $7.6 billion in the US, and it's only continued to grow. Texas is kind of an economic powerhouse in terms of agricultural production for both cotton and peanut, both of which are in high demand for organic products. And Texas is the only state to produce all 4 varieties of peanut plus organic peanut. However, the implementation for organic management is very, very time and cost intensive. For example, the USDA requires 3 consecutive years of organic management prior to allowing farmers to sell their products organically. And so that is an economic loss during that period of time. And in terms of the practices required for organic management, no GMOs are allowed, no synthetic fertilizers are allowed, and there are heavy restrictions on what can be used for herbicides and pesticides, all of which can result in depressed yields, especially during the first few years of implementation. However, there are specific practices that are highly suggested to use in tandem with organic management, which have the goal of improving soil health and thereby improving yield over a long period of time. But very few studies have focused on organic management specifically for these management practices within the first few years of implementation. One such management practice, which is associated with building soil health and is highly recommended for organic farmers, is cover cropping. This is assisted, as I mentioned, with improved soil health by reducing wind erosion, building up soil organic matter over time, and thereby stimulating microbial activity and the nutrient cycling associated with that. However, there is a national reluctance to begin using cover crops, and in Texas, this can be due to concerns over excess water usage and cost, as even though cover crops have been shown to provide benefits for the soil, it's not always easy to predict the benefits, especially in terms of the economic benefit. And although there have been studies showing that, yes, you can get improved yield when using cover crops, again, this isn't always predictable. So that, coupled with the wide range of cost for cover crop seed, can turn farmers away from using cover crops. And so my portion of the study focuses on whether or not there is a cover crop that early on in implementation provides strong benefits for soil health. So my objectives for this study were threefold. Firstly, to determine how soil health reacts to a recent change in management to organic agriculture. Secondly, to determine how soil health is impacted by a recent cover crop implementation. And finally, to determine how microbial activity is influenced by cover crop seeding rates in organic agriculture. Due to the time limits and limited significant results that I have found in the final bullet thus far, I won't be addressing that today, and I will only be addressing the second bullet to a limited extent. This study was located at the Lubbock and Vernon AgriLife Research Centers. Special thanks to Dr. Katie Lewis and her crew for managing my plots at Lubbock, and to Dr. DeLon and his crew for managing my plots at Vernon. The Lubbock location represents the High Plains ecoregion of Texas, while the Vernon location represents the Rolling Plains ecoregion. And they are both considered semi-arid regions, receiving 15 to 20 to 20 to 28 inches of annual rainfall. My plots, the soil type of my plots specifically at Lubbock was considered an Olton clay loam, while at Vernon, my plots were a Miles loamy fine sand. The study design was a randomized complete flock for both organic and conventional agriculture. The organic plots received organic cover crop seed and mechanical cover crop termination and weed control. So there was a little bit of light tillage early on in the season, especially. And the organic plots also had compost and manure in place of chemical fertilizer. Both treatments, however, received cover crop treatments that included rye, radish, rye vetch mixes, and rye vetch radish mixes. Of note, the vetch is a legume, so that was also fixing nitrogen in the system, and I'll be referring back to that a little bit later as well. For sampling, I collected monthly composite cores at the 0 to 4 inch and 4 to 8 inch depths. These were air dried for testing. And I also collected monthly respiration data using a GasMet DX 4040 FTIR. This was hooked up to a LICOR soil chamber and PVC soil collars to collect respiration data specifically on CO2, but also for nitrous oxide and methane throughout the course of my study. At the beginning of the season, prior to cover crop termination, we collected cover crop biomass from the 2nd and 4th rows, and we also collected peanut yield at the end of the season. Finally, we also had a bit of an exploratory portion of this study that focused on nodulation, and that data was collected in August when peak nodulation is expected to occur for peanuts. So my timeline, as you probably can tell by now, we had a cotton peanut rotation for both of my sites. This was established in 2019 with the cotton rotation, and it had also been established at the Lubbock location for, I believe, 5 years prior. So in 2020, when I began my collections, both sites were planted to peanut, and that was variety ACI 236. The study for me really began in late March to mid-April. March was when cover crop termination began, and that did extend through April a bit because the conventional cover crops at Lubbock were resistant to the Enlist duo that was used to terminate them, and so a couple rounds of Roundup were also required. In May, fertilizer was applied to Vernon fertilizer was not applied to Lubbock because in general, when you're working with peanuts, since they're legumes, and since they do need to associate with their bradyrhizobia for effective nitrogen uptake, you generally want them to be under a somewhat of a nitrogen-starved environment. So it's recommended to apply any kind of fertilizer the previous rotation. However, for Vernon, fertilizer was not applied during the cotton rotation, and so it was applied during right before the peanuts were planted. And I do believe that this had some effect on my results, so I will also be referring back to that multiple times. Peanuts were planted afterwards in May. And then in June, we discovered that there was poor initial growth from our Lubbock plots, and so a replant was required for Lubbock. So the peanuts were about a month behind the Vernon plots. In August, we had our nodule collections, as I mentioned, and then mid-October was when harvest occurred, followed by my final sampling. Again, I had been doing sampling each month up until then. The testing that I did for my study was on mineralizable carbon, enzymes, and PLFAs. Of note, the PLFAs were sent out to testing labs just because of the extensive nature of testing for that. Mineralizable carbon was used because this is considered a sensitive measure of change in soil health according to management, and it generally represents microbial necromass or decaying organic substrates within the soil. This test is done via a 3-day incubation where a set amount of soil is introduced into an incubation jar with a NaOH trap. They are incubated together for 3 days, and afterwards the NaOH trap is titrated so that you can back calculate the amount of carbon that was evolved from that soil during that time. For enzymes, the reason why we used enzymes as a measure of soil health was because enzymes themselves, their goal, or the goal of whatever organism, the goal of using enzymes is to reduce the amount of energy required for a reaction to occur. And so microbes, especially in soil situations, tend to use enzymes to catalyze the release of simple sugars that they can then use as a source of energy. The 2 enzymes that I focused on were beta-glucosidase and N-acetylglucosaminidase. Beta-glucosidase is involved in the last step of cellulose breakdown, and it releases simple glucose monomers, again, for microbes to use as energy. N-acetylglucosaminidase basically does the same thing, except it acts on chitin, which is in the cell walls of fungi and insects. And it releases amino sugars, and the nitrogen within those amino sugars can make up anywhere from 5 to 10% of organic nitrogen in the soil. But this is also dependent on the carbon to nitrogen ratio in the soil already. And calorimetric assays were used to measure the enzyme activity of both of these enzymes. For PLFAs, the goal of the PLFAs are phospholipid fatty acids. And basically, this test looks at what the general microbial community structure is, and it uses the fatty acid chains associated with certain microbial groups to categorize what kinds of microbes are present and in approximately what quantity they are present. So again, these were sent out to testing labs for obtaining these results. We've received the data from the Lubbock location, or from the Vernon location, but not the Lubbock location yet, and so I can only provide a limited amount of data on that. All right, and now we can get into our results. I'm going to begin by discussing the cover crop biomass harvested from both the organic and conventional trials at each location. Of note, at Lubbock, the organic plots had significantly greater cover crop biomass compared to the conventional, and this was flipped at Vernon. I will be referring back to this later on because when you leave cover crop residues on top of the soil, this can affect microbial activity as they will be using enzymes to break down these residues. And so that will have some degree of effect on the data that I show you in a bit. For PLFAs, it's interesting at Vernon because this data was collected prior to fertilization. And you can already see that for a number of the categories of microbial communities, greater PLFAs were found under organic management as compared to conventional management. This was only significant for a limited number of these PLFAs, but it is still interesting to see that difference. In terms of cumulative CO2 flux, this was collected with the gas MET that I mentioned earlier on, and it generally represents respiration throughout the course of the growing season. I've categorized my conventional treatments as shades of red and my organic treatments as shades of blue. And overall, you can see that plots that received a cover crop did generally have greater respiration throughout the course of the season as compared to the fallow, which is in black. One thing to note was that at Lubbock, there was a cover crop failure for radish, and those would be the red and blue lines that are most in sync with the fallow. But in general, greater respiration occurred under cover cropped plots, especially cover crop plots that were under the conventional treatment. A similar trend occurred at Vernon. However, this did not result in significant data. In terms of mineralizable carbon at Lubbock, throughout the course of the growing season, in general, you could see that greater levels of mineralizable carbon were available under conventional plots as compared to the organic. And this was most significant at the 4 to 8 inch depths. So we have April, May, and July and August all have significant differences between the organic and cover crop and conventional plots. But at Vernon, this was somewhat flipped, especially early on in the season up until July, where you can see that there was significantly greater levels of mineralizable carbon under the organic plots as compared to the conventional, in particular at the 0 to 4 inch depth. What this tells me is that the manure and compost that was applied was probably affecting these results, especially because manure and compost has a lot of decaying organic matter in it, and that is going to very likely show up in this mineralizable carbon test. For the enzyme activity data, I'm only going to be presenting the mid-season data for right now, just because the data set for the pre and post-season is as of yet incomplete. But in general, for both locations, you can see that there's a trend towards greater enzyme activity under the organic plots. At Lubbock, this was only significant at the 4 to 8 inch depth for N-acetylglucosamidase, but there is definitely a trend towards it at the 0 to 4 inch depth for beta-glucosidase, likely related to that greater level of cover crop biomass that I had mentioned earlier in these slides, cover crop biomass for organic. And at Vernon, at both depths and for both enzymes, there's significantly greater levels of enzyme activity under organic. Again, I believe that this is related to that compost and manure which was applied, which, like I mentioned, has a lot of organic material for microbes to act upon and use as energy. All right, and now I'm going to be presenting a little bit of my nodule data. Remember, this was an exploratory section of our project, and my expectations for this had been that I would have greater overall nodulation and percent active nodulation under the organic rye. I expected this because, as I mentioned, peanuts or legumes in general tend to nodulate best under somewhat of a nitrogen-starved condition, and the organic rye would not have received synthetic fertilizer, and it would not have received that nitrogen fixation from the vetch. So the Vernon location does fulfill my expectation, but the Lubbock location, oddly enough, does not. For some reason, there is significantly greater levels of total nodule counts and percent active nodulation under the conventional plots as compared to the organic. It's also important to look at this data in terms of total nodule counts per centimeter squared first. I did have to normalize this data according to taproot area, just because the root lengths that I collected varied wildly. And so at Vernon, we collected a massive number of nodules, but very, very few of them were actually active. At Lubbock, the opposite was much more true, where far, far fewer nodules were collected, but the vast majority of them were active. And you can see that in these two pictures to the side. The top picture represents a root from Lubbock. I have a couple nodules cut open, and you can see that they are bright red inside, indicating that they are active. For Vernon, you can see that is the picture below. There are a ton of nodules on it, and I have one of them cut open, but you can't really tell which one it is because it's not bright red inside. It's kind of like this off-white, almost grayish color, and that is this one that I have my pointer over. And then finally, we have our peanut yield data. At both locations, there were no significant differences between treatments according to management. And I also did not really see many significant differences according to cover crop treatment either, which is significant for farmers looking to take the economic risk of switching to organic management, because this tells us that at least for this year and for this crop, there was not a yield loss when using organic management. So overall, at this point in time, it's likely too early to really see many significant differences according to cover crop selection, which is one reason why I didn't include much of that data. But there were a few instances where p-values began to approach significance. So later on, as this study progresses, we might start to see some of those significant differences according to cover crop selection. But it is significant that even now, we can already see differences in cumulative respiration according to whether or not a cover crop was used, and to some degree, whether or not that cover crop was in organic or conventional management. Also of note, for this year and for this crop that we used, we didn't see any negative impact on yield based on cover crop used. And then we do see some potential early indications of change in soil health according to management practice. For example, the elevated PLFAs under organic management, pre-fertilization at Vernon. And for mineralizable carbon, there are definitely some significant differences according to treatment. At Lubbock, the conventional treatments resulted in elevated mineralizable carbon throughout the growing season, while at Vernon, this was somewhat flipped, likely due to the application of fertilizer and manure. And the potential indication of elevated enzyme activity under organic management at the mid-season was also an interesting indication of potentially early change in soil health. It is also interesting to note the nodulation patterns. To some degree at Vernon, they did follow through with my expectations, but at Lubbock, some of the results were against my expectations. And it will be interesting to look at nitrogen data that we'll be getting in the future to see if there is any nitrogen availability results that could explain what happened with nodulation at Lubbock. Finally, it is definitely of note for farmers that we did not see a yield loss due to organic management within the first year of this study. I would especially like to thank my graduate committee, Dr. Katie Lewis, Dr. Terry Gentry, Dr. Paul DeLon, and Dr. Elizabeth Pearson, and extend a special thanks to the Lubbock and Vernon crews and my fellow graduate students for providing an immense amount of support throughout the course of this study. Finally, I would like to thank the Southern SARE program for funding my research. Thank you.
Good job. Great job. Very interesting dirt work. Now, any questions?
I see a few coming in. Okay, 1 second.
Hey, you know, you guys can ask your own questions. You don't have to write them in the chat. Just pipe up.
Yeah,
All right. Yes, who is it?
Vinita, and I enjoyed your presentation, and I wanted to know about your plot. What was the history of your plot? Was it already your plot maintained as the organic block, or you just use for your research only?
Yes, so this was the first, 2019 was the first year where organic management had been implemented at both locations. And at Lubbock, the plots that I used had been previously planted to that, the peanut and cotton rotation for, I believe, 5 years. At Vernon, that was the first year of cotton peanut rotation. But to answer your question specifically, 2019 was the first year of organic management.
Okay, thank you.
Mm-hmm. Should I go to some of the questions that
Let's have them ask it out loud. Yeah. Pipe up.
I can.
Hi, Lydia, can you hear me?
Go ahead.
Yes.
So I have a question about percent organic matter in the soil due to the addition of cover crop. Do you actually see an increase in percent organic matter in the soil samples?
So I don't have that data yet. We are going to be getting more data on organic carbon, and that will be representing the pre-season, mid-season, and post-season. But we're still waiting on that data.
Okay. And then lastly, can you talk a little bit about the variations they see in your data? Because you don't seem to have a lot of standard deviation bars.
Yeah, so let's see.
I mean, just kind of in general, do you actually see your data to be pretty consistent among your replicates, or do you actually see a pretty big variation?
That really depends on the cover crop used. The data that I presented was for the organic plots that would have included all the cover crops under the organic, and for the conventional plots that would have included all the cover crops under the conventional. And so there wasn't a huge amount of deviation just because I had that much more data. But when I'm looking at things in terms of cover crop treatment itself, then variation is a fair bit greater, just because then I'm only looking at 3 reps as opposed to a much larger sample size.
Okay, thank you.
We have time for one more question.
All right.
Hi, Leah. I have a question.
Yes.
So I have a question on the design of your experiment. So you had your treatment management practice as organic and conventional. Mm-hmm. So how did you assign the treatments? Did you assign randomly organic and conventional in each block, And if so, then also, if it is RCBD, how did you assign the cover crop treatments? So is it like the organic and conventional plots were like all side by side in a block in a randomization?
So the way that we did this was we had a large block of plots that were just organic, and that block of plots was a randomized complete block with all the different cover crop treatments in it. Nearby, about, let's see, I want to say maybe less than 50 feet away. Dr. Lewis, if I'm wrong about that, correct me. But very close by was a second block of plots that were all conventional. And within that block of plots, it was a randomized complete block for each of the cover crop treatments.
So, oh, I got it. So you had like RCBD in organic and RCBD in conventional, right?
Yep.
And one more question. So how did you measure the CO2 flux in the field? Did you have had like chambers installed in the field?
Yeah, so what we did was when you use the gas mat, at least the way that we use it, we have these PVC pipes, all of which are the same size, same diameter. We place them out in the field for 24 hours to allow for soil disturbance to not affect our results. And then we go out into the field with a LICOR chamber that we fit on top of those PVC pipes. That LICOR chamber is connected up to our gas met, and then the gas met can measure, basically, once you turn the gas met on and start measuring, the air that is under the chamber and the PVC pipe just cycles through the gas met, and it takes a reading on ppm of each gas, like every 20 seconds, I believe. So you can look at the gas flux that way. And we also take measurements of how deep into the soil we put our PVC pipes so that we know exactly what the volume inside of the PVC pipe and chamber is. And that allows us to do the measurement.
I got it. I just didn't notice that you mentioned gas mix. So.
Oh, yes. Mm-hmm.
Yeah, I got my answers. Thank you.